Please welcome Professor Nancy Yuk-Yu, President, the Morningside Professor of Life Science, Director of the State Key Laboratory, Molecular Neuroscience, the Hong Kong University of Science and Technology, and founding member of the Hong Kong Academy of Sciences, to come on stage to deliver her keynote speech. The title of the speech is Circulating Proteins, Potential Game Changers in the Diagnosis and the Treatment of Alzheimer's Disease. Professor Yi, please.
Thank you. I want to first thank LabChi, Harry, and organizers for inviting me to this exciting symposium. I greatly appreciate this opportunity to share with you the recent findings from my lab on circulating proteins, which we believe are the game changers in the diagnosis and treatment of Alzheimer's disease. With the aging population worldwide, including Hong Kong, there's an increase in prevalence of age-related degenerative diseases, such as Alzheimer's disease. Now, the most common form of Alzheimer's disease is the late onset AD. And it has been projected that by the year 2050, there will be more than 150 million people worldwide who are afflicted with this disease. It's also estimated that half of the individuals aged over 85 will suffer from the disease. As you know, AD is an incurable and progressive neurodegenerative disease. I suppose we can view this as an invisible pandemic. The patients suffer from memory loss, faulty reasoning and judgment, as well as impairment in locomotor ability. The hallmark of this disease is the presence of amyloid plaques and neurofibrillary tangles in the brain. Individuals that have mild impairment in cognitive function, they are at high risk to progress to Alzheimer's disease. For the AD patients, when they have memory problem, they will consult the clinician. But by that time, they are already at a rather moderate or advanced stage of the disease. In fact, there are pathological changes that occur in the brain 10 to 20 years before the clinical symptoms happen. Currently, there's a lack of objective diagnostic tools. There's also a lack of effective interventions and treatment. So there is an urgent need for us to develop early diagnostic tool and also more effective interventions. The earlier is the diagnosis, the earlier can one have early interventions and hence more effective disease management. Now, we all believe that development of biomarkers is critical for AD diagnosis. With the advent of transformative technologies, we now have new AD biomarkers, including genetic, including gene transcript and protein expression, as well as brain imaging. Now, I believe that the identification of biomarkers can greatly enhance the diagnostic accuracy and also stratification of patients. And this would lead to more effective diagnosis and also monitoring the response to disease-modifying treatment. I also believe that research based on biomarkers can help us identify novel disease targets for the disease and hence develop new strategies for identifying therapeutic treatment. Now I want to review with you the current AD diagnosis. They include clinical assessment by clinicians to take family history for the individuals to do a written assessment. But this, I'm afraid, is too subjective. They are not objective enough. Another way of diagnosis is through brain imaging, for example, the amyloid PET imaging. But these are very expensive. And more recently, there's also development of biomarkers in the cerebrospinal fluid. But this is considered rather invasive. Hence, we believe that it is important and also urgent for us to develop blood-based biomarkers. For a while, it's believed that the pathological hallmarks can be detected in the blood, but these are very few protein biomarkers. But if we can develop a blood-based biomarker that is convenient, simple, and less invasive, they will have great potential for development. In order to achieve this, we have to first identify the blood biomarkers for AD. So my lab undertook a comprehensive study to identify the biomarkers that are associated with the disease and the progression. And how did we achieve this? We used the most advanced blood protein detection technology that gave us the sensitivity that we need, because many of these proteins, they actually express at very low level in the blood. We also leveraged the high throughput big data analysis. So with these two technologies, we were able to overcome the challenge. We were able to overcome the bottleneck of the current blood testing technology and hence achieve the testing using small sample volume, a test that has high sensitivity and high throughput. So we first conducted a comprehensive screening of blood proteins in order to select for blood biomarkers with the characteristic of AD pathogenesis. This idea was very novel at that time because it was widely believed that one would not be able to identify so many proteins in the blood that are characteristic of the disease. So we first measured the levels of over 1,100 proteins in the blood, and we compare the level in AD patients versus the healthy control. Then we analyze which of these proteins are being altered in the AD patient. And using the big data analysis, we were able to come up with 19 proteins that we believe are the blood protein signature for the disease. Furthermore, we developed a scoring system that will allow us to do early screening and also staging of the disease with an accuracy of over 96%. So out of the 1,100 proteins we assayed, we observed that more than 400 of these proteins were changed. Actually, a large number of these proteins were downregulated. And with this data, we further performed co-regulation analysis. We try to understand which of these proteins are co-regulated together. And through that kind of analysis, we identified 19 of such protein clusters. Within each protein cluster, we identify a hub protein. So the hub protein is representative of that particular cluster. So altogether, we came up with 19 hub proteins. We then use these proteins, the 19 protein biomarker panel, and compare with the so-called ATN panel. So I mentioned earlier that the pathological hallmarks of the disease include amyloid and tau. So the ATN panel was developed not too long ago, and it was used as the blood biomarker test. But our blood biomarker panel with 19 proteins, when compared with the ATN panel, ours actually gave us better result. Then we use our 19 protein biomarker panel to analyze the AD patients. We found that we were able to diagnose the disease, and also we were able to stage the disease into mild and severe, and compared to the healthy control. We obtained very good association with the cognitive performance. So the score that we obtained, or the staging of the disease we were able to observe, actually correlate very well with the cognitive performance as reflected by the MoCA score. It also correlates very well with the hippocampal volume. So we believe that this panel can be applied to staging and monitoring the disease progression. Now, this particular diagnostic tool that we have developed is highly sensitive and specific. It can provide us with the tool to perform early diagnosis, which would give us better prognostics. It would also enable the continuous monitoring of the disease status. And finally, we can use this to screen suitable individuals for the clinical trials, and also be able to monitor the efficacy of the therapeutic treatment. So we are currently developing a more cost-effective blood test so that we can do large-scale screening. Now, just now, I focus more on the development of blood tests based on the biomarkers. I now want to shift to the second part of my talk, which is whether these plasma proteins actually can be developed as therapeutic targets for AD. Now, as you know, AD is a very complex disease. With the accumulation of A-beta, it can trigger the immune cells in our brain called microglia. Microglia can phagocytose A-beta, hence resulting in its clearance. But as A-beta continues to accumulate during the progression of the disease, microglia became overwhelmed, hence resulting in the dysregulation of this cell type and chronic neuroinflammation, ultimately leading to deficits in synaptic remodeling and neurodegeneration. So pathogenesis of AD actually involves many cell types, and I highlighted a few of them here. It includes neurons, microglia, astrocytes, endothelial cells, all these different cell types in our brain. It is important for us to identify the molecular and cellular mechanisms that can contribute to the disease pathogenesis. The one pathway that we were particularly interested in is the one mediated by the cytokine IL-33. IL-33 acts as an alarmin to maintain the immune homeostasis of our body. This cytokine mediates the cellular response by acting on this heterodimeric receptor complex, ST2 and IL-1 receptor accessory protein. ST2 receptor also occurs in a soluble form, and it serves as a decoy receptor. And by binding to IL-33, it can actually result in the inhibition of the normal function mediated by IL-33, and in doing so, it can be considered as an endogenous inhibitor. When we look at the level of soluble ST2 in the plasma of AD patients versus healthy control, we were very intrigued to observe that in the AD patients, there's a higher level of soluble ST2 in their plasma. Furthermore, we also observed that in the cerebrospinal fluid of the AD patients, the soluble ST2 level is also elevated. Now, when we look at the A-beta plaque load, remember, A-beta plaque is the pathological hallmark for the disease. We found that the higher the level of soluble ST2, the higher is the A-beta plaque load. So what that says is that high level of soluble ST2 perhaps play a role in the deposition of A-beta. So we did a lot of experiments using animal models, and I just want to show you two pieces of data to share with you what would be the consequence if you increase the level of soluble ST2 in the mice. And we did that by injecting soluble ST2 into the brain of those mice, injecting them using the ICV approach, that is, into the cerebral ventricles. When we compare the mice that got soluble ST2 injection versus the mice without, we found that the mice that got soluble ST2 injection, they actually have a much higher A-beta plaque load. So what that means is that if you increase soluble ST2, you will actually exacerbate the pathological changes in the brain of those mice. Now, I mentioned to you earlier that microglia is the cell type that phagocytose A-beta and clear A-beta. So we therefore use a fluorescent dye called methoxyXL4 to label the A-beta, followed by flow cytometry analysis. And we look at the microglial A-beta phagocytic ability. That is, when the mice have more soluble ST2, what would happen to the ability to phagocytose or eat up the A-beta? We found that the mice that receive soluble ST2 injection, they have an impaired ability to phagocytose A-beta. So this observation is consistent with what we observed on the A-beta plaque load. That is, the higher the level of soluble ST2, the worse is the ability of the microglia to eat up A-beta, and hence the higher is the A-beta plaque load. So elevated level of soluble ST2 plays a very important role in AD pathogenesis. So is this circulating protein, soluble ST2, a good target for AD? So we conducted a number of experiments to try and understand how is this circulating protein regulated and whether it can contribute to the pathogenesis of the disease. And for this, we use advanced next-generation sequencing technologies. So first, we perform whole genome sequencing of AD and healthy control in Hong Kong. And then we performed the genome-wide association analysis. For those individuals that have known data on the soluble ST2 level, through the GWAS analysis, we were able to identify genetic variants at a particular locus, as shown here. IL-1 receptor L1 locus. This particular locus is what modulates the level of soluble ST2. We further identify a genetic variant highlighted here. This particular genetic variant, if the individuals carries 2 copies of them, AA, they actually show a reduction in the level of soluble ST2 in the plasma. So what that means is individuals that carry this genetic variant, they will have lower level of soluble ST2. Now, what is the brain cell type that make this soluble ST2? We perform the single nucleus RNA sequencing of human brain samples, and using this particular marker, CLDN5, which marks the endothelial cells, we were able to identify the human brain endothelial cells as the major source of soluble ST2. Furthermore, we look at the level of soluble ST2 for the individuals that carry this variant, and we were able to show that indeed in those individuals, the level of soluble ST2 is reduced. So the AA carrier has lower level of soluble ST2. So this is a very important finding because it shows that the genetic variant that we have identified is associated with decreased soluble ST2 gene expression in the endothelial cells. Now, what would happen if we delete that particular region, that particular genomic region that harbor the genetic variant? For that, we turn to CRISPR technology to help us delete the specific regions that harbor the genetic variant. And we found that when we delete that particular genomic region, the gene expression of soluble ST2 is abolished. So it shows that the genetic variant that we have identified is a key genetic factor that regulates the soluble ST2 level. I don't want to go into details of this analysis, but suffice to share with you that this particular allele exerts a protective effect against AD risk. So how does it play a role in AD pathogenesis? We found that individuals that carry this protective variant, they have a delay in the onset of the disease. So comparing AA with the non-carrier, it also shows an increase in the size of the entorhinal cortex. These are AD patients. Moreover, it can also protect against the brain atrophy, the brain shrinkage that is characteristic of AD individuals. And this study was performed in collaboration with the Australian group that has followed these individuals for 7.5 years. So the carriers, the protective carriers, actually has a slower decline in terms of the brain volume. Now, it is very important for us to figure out the mechanism. So again, by doing these single nucleus RNA sequencing analysis, we found that the individuals that carry this protective variant, they actually exhibit activation of the genes in the microglia. Remember, I mentioned earlier that microglia is the cell type that can phagocytose A-beta, resulting in A-beta clearance. So the fact that the individuals that carry this protective variant show activation of these genes that can mediate A-beta phagocytosis, therefore reveals the underlying mechanism why these individuals show the protective effect against AD. And finally, we perform additional studies to show that for individuals that carry this allele, there is increased interaction between the plaque and the microglia. So this purple is microglia, the brown color is the plaque. So there is an increase in the interaction of microglia and the plaque, as shown here. And this is what led to the reduction of the A-beta plaque load in those individuals that carry the genetic variant. So just a summary slide to share with you. The cytokine that we look at, IL-33, acts on the microglia to trigger A-beta phagocytosis and hence the clearance of amyloid. During the disease progression, there is increase in the release of soluble ST2, which acts as a decoy receptor to inhibit the beneficial action of IL-33 and hence resulting in increased deposition of A-beta. We have identified a protective genetic variant that is associated with reduction of soluble ST2, and it can protect against AD risk. So this suggests that it is a good target for us to develop strategies for AD intervention. It is also very important for us to use this to stratify patients for therapeutic treatment. And my lab is actively looking at development of modulators for soluble ST2. So I want to close the talk by thanking the talented students and staff in my lab, and also my collaborators that provide us with the clinical cohort, and my funding agencies as well in Hong Kong and in the mainland. Thank you very much for your attention.
Thank you, Professor Yip. Please be seated.